Hello, my name is Christoph Scherber. I'm from the Leibniz Institute for the Analysis of Biodiversity Change in Bonn, Germany. Today I would like to talk with you about hardware and software tools to assess agricultural biodiversity. There are several challenges in assessing biodiversity in farmland. For example, measuring what's happening inside the middle of agricultural fields. And there are trapping devices such as the yellow pen trap that you can see in this picture that can help collect biodiversity data from inside the fields. In addition, another challenge is to unravel the network structure of agroecosystems. You can see here, for example, management practices and how these affect interactions among organisms and ultimately economic output such as yield. And unraveling these structures can be done, for example, using different sensor systems. You can use basically 4 different sensors to assess agrobiodiversity. One of them being optical sensors such as camera traps, but also acoustic sensors using microphone arrays, chemical sensors, for example, to measure volatile organic compounds, or molecular analytics using DNA metabarcoding. Optical sensors may be most familiar to you, one of them being the typical wildlife cameras here on the left-hand side, but also time-lapse cameras such as this one here that take shots every time interval and that can then be used, for example, to measure insect occurrence in farmland. A new development is the so-called dynamic vision sensor that can also aid in identifying components of biodiversity such as insects in a particular image. The trick being that also information on the movement of objects is recorded, and this information is used to classify objects. Extracting information from images taken in the field can be an important task. You start with classification of the image, whether it contains an insect or not. Then you detect the insect by a bounding box, and then you use segmentation tools, image segmentation tools, to find out more details about which kind of insect is present. Similar approaches can be used using acoustic sensors, this one being a sensor in a little plastic box here that is waterproof. And you get acoustic signals, such as calls of birds or bats, out of it. We are collaborating, for example, with the Fraunhofer Institute to disentangle different call activities and different species. This is an example of what can be done. In acoustic detection, you do a spectrogram analysis, and you can find out about different bird species that are calling over a given period of time in a sample. You can find more details about this in this paper by Wolfgang Wägele et al. That is just impressive, basic and applied ecology. Finally, you can use automated insect traps, such as these ones here. These are automated malaise traps where the insects fly in here and then they end up in a sampling vial. These sampling vials are then rotating at a given period of time, and you can extract information on which insects are present at which point in time. This is an example for what you get out of it. You can use DNA sequencing machinery on the bulk sample, and you get a whole range of information on the taxonomic composition and biodiversity of this sample out of it. Finally, you can also go for trophic interactions. Who is feeding on whom? You can find out what this beetle, for example, is consuming using the same methodology. You collect the regurgitate, you use particular primers, and then you sequence what's happening here, and you find out about trophic interactions of this beetle with other organisms. And again, I refer you to a little paper down here by Tide et al. 2016, where we showed how this would work in principle. You can then use high-dimensional data analysis to find out which of the many species in a sample are responding to which drivers, and you get estimates of the coefficients for each species in that sample. We are currently applying these methodologies hardware and software tools in the following 2 projects, one being the plant teams project here, Diversify, an EU project where we use these kind of interval cameras to identify insect presence in different cropping systems. And we are doing following similar approaches in the Finca project, where we have a total of 90 fields and we measure insect numbers and activity using a range of techniques. With that, I would like to thank you very much. And if you're interested in further details, check out our homepage here at the Leibniz Institute ZBM, or just drop me an email. Thank you very much for your attention.